So let's begin looking at the transport of the gases, carbon dioxide and CO2 in the blood. Our third objective to describe the transport of different gases, mainly O2 and CO2 in the blood. And here we want to think about the structure of hemoglobin. Oxygen is transported by hemoglobin in the blood. We described earlier when we looked at um, the solubility of oxygen and compared that to the solubility of CO2, we said that CO2 is much more soluble in blood, oxygen not so much. Okay, CO2 is actually twice as much soluble um, in liquid, specifically in our plasma, than CO2 is, or excuse me, than oxygen is. So only about 1.5% of arterial blood oxygen is moved around, freely dissolved in our plasma. The rest of that, most of that oxygen has to be bound to hemoglobin in order to be transported, right? It's not very soluble in terms of just being transported freely in our plasma. And so that is the role of hemoglobin. This is why we have erythrocytes. Again, they have no other machinery. We talked about how they don't have a nucleus, not much other organelles within those cells. All they have is hemoglobin, which has the ability to bind four molecules of oxygen and transport those oxygen around in our blood. So that's the sole purpose of our red blood cells. When we think about the state of hemoglobin, when it has oxygen bound, it's going to be called oxyhemoglobin. And when it doesn't have oxygen bound, it's going to be called, I should say fully saturated, it's going to be called deoxyhemoglobin. So oxyhemoglobin is that fully saturated bound oxygen, and deoxyhemoglobin is not. So having said that, we can look at the equation. The equation which goes right to left or vice versa with deoxyhemoglobin and oxygen. On the other side, we get oxyhemoglobin, and then we can have that equation occur in the opposite direction depending on the saturation, depending on the levels of oxygen on either side of this equation. And then looking at the illustration here, here is our alveolus in the lungs. We know that the partial pressure of oxygen here is 100 millimeters of mercury. In the pulmonary capillaries, it's 40 millimeters of mercury. So we have a gradient for diffusion of our oxygen. And the way that diffusion occurs is across our respiratory membrane. Right? Don't forget the structure here, crossing these two cells and their fused basement membrane, and then going into the erythrocytes that are in the pulmonary capillaries, and then binding onto the hemoglobin inside those erythrocytes, and then being transported on those hemoglobin. In terms of the systemic capillaries, similar exchange is happening in the opposite orientation. So the oxygen is leaving the hemoglobin on the erythrocytes, crossing our our tissue capillary beds, and then diffusing into our cells at the tissue capillaries. And if we think about our gradients here also facilitate that diffusion. So the partial pressure in the arteries at 100 millimeters of mercury, and then here at the cells of the tissue, 40 millimeters of mercury, which facilitates the diffusion from the arteries into the cells. Now, in terms of the state of hemoglobin, we can think about it as being fully saturated when it's carrying four oxygen molecules bound onto those heme binding sites. So it is fully saturated when it has all four sites bound. Now, in terms of the law of mass action, we've described this in other areas in terms of driving our equation to the right or to the left, depending on the amount of oxygen available. So the more oxygen we have, the more we're going to drive the equation to the right to create oxyhemoglobin. All right, if we have lots of oxygen available, we're pushing the production of oxyhemoglobin as there's more oxygen available to be bound, and then we have complete saturation of our hemoglobin. Okay. If we had lower oxygen levels available, let's say again we had states where you are using oxygen faster than it's being supplied, you would have lower oxygen available and you would drive your equation to the left, which is towards the deoxyhemoglobin state. Now, this gradient is also important in terms of unloading and loading oxygen. If we have more oxygen available and we have less saturated oxyhemoglobin, what we're going to find is that we drive the equation towards the loading of oxygen, which happens in the lungs. In the tissues, we have less oxygen, which drives our equation to the left towards the unloading or the offloading or the delivering of oxygen towards that tissue. Okay, so in this state, it's 100% saturated with 4 oxygen bound to those hemocytes. Let's look at the oxygen carrying capacity of our blood. So when we have fully saturated hemoglobin, that's about 1 gram of hemoglobin, which is carrying about 1.34 milliliters of oxygen. So this is when hemoglobin is fully saturated. And in terms of that capacity, we can look at the state to the panel A here. So all 4 sites have oxygen bound, so it's fully saturated here. In terms of the hemoglobin levels, the normal hemoglobin levels in our blood, about 2 to 17 grams per deciliter, and this can be slightly higher in women than it is in men. In terms of the oxygen carrying capacity of hemoglobin, it's about 200 milliliters of oxygen per 1 liter of blood. All right. Now, the arterial blood is going to have hemoglobin at 98.5% saturated, which is very close to 100. We're not going to say fully 100, just because a few of these may not be all the way saturated, as we can see here, but very close to being 100% saturated. And then in the venous blood, when it is 75% saturated, we only have 3 oxygen molecules on the hemoglobin. Only 3 of those sites are bound in our mixed venous blood, which is at about 75% saturation. And this is when the partial pressure is about 40 millimeters of mercury. Now, what this means overall, like all of this to say that the removal of 1 oxygen molecule from hemoglobin is all that it takes for the partial pressure in hemoglobin and the partial pressure in the tissue to be at equilibrium, and for our hemoglobin to be 75% saturated and no longer able to offload any more oxygen. And what this really means is that we never really offload all of the oxygen from our hemoglobin. Why? Because that gradient is so essential to the offloading and onloading states that all it takes is the offloading of 1 oxygen per hemoglobin for our tissue levels and our hemoglobin levels to be at equilibrium, and we no longer have diffusion of those gases. Okay, so the removal of 1 oxygen is all that we need for our partial pressures to be equal and to no longer have a gradient to facilitate any further removal or diffusion. So here is our oxygen dissociation curve. This is a very important curve that shows the way oxygen is offloaded at different states, different places in our systemic veins, in our systemic arteries. So this is the percent oxygen saturation, right, from 0 to 100. And you can think about this as the number of oxygen molecules that are bound to hemoglobin. And so we can think about this in fourths. So 25% is 1, 50% would be 2, 75% would be 3, which we know is our state in the systemic veins. And then 100% would be all 4 hemoglobin sites having oxygen bound. So if we think about the systemic veins, they're at 75% oxygen or 75% saturation in terms of having 3 out of the 4 oxygen molecules present on that hemoglobin. And then between our systemic veins, where we have 40 millimeters of mercury being our partial pressure, and our systemic arteries, where we have 100 millimeters of mercury, and we have all four oxygen binding or oxygen sites bound, now we can see that this is where we have the loading and unloading area between our hemoglobin molecule. So this is where that last oxygen is bound or offloaded. It is bound between the veins and the tissues, and then it's offloaded at the systemic capillary. So it's, let's say that again, it's bound in the pulmonary capillaries between the systemic veins and systemic arteries, and then it's offloaded in the systemic capillaries, again, between systemic arteries and systemic veins. And then we can compare that between the number of oxygen bound and the partial pressure of oxygen in that state. So we can also see that at the 75% saturation, which is where we have 3 oxygen bound, our partial pressure is 40. So it's going to be the same in the cells as it is in the systemic vein. So there's no diffusion, reiterating that idea that as soon as we remove one oxygen molecule, we're at equilibrium. No longer have a gradient, no longer have any movement of gases for any further offloading of oxygen. And then the partial pressure in our arteries at 100 millimeters of mercury, okay, this is where we have all 4 oxygen molecules bound. Now we have a gradient which facilitates the unloading. Okay, we can also think about the oxygen dissociation curve shifting to the right or shifting to the left, meaning that it becomes either easier or harder to offload those gases, okay? In the case of the right shift, this is the first scenario that we will consider here. This is called the Bohr effect. This is the effect of the pH on the oxygen hemoglobin dissociation curve. And basically at lower pH levels, let's say someone has a lot of acid in their blood, or they have a lower pH in their blood for many different reasons, that actually increases the oxygen unloading. And in our active tissues, we're going to see that there's more acid produced. The pH decreases, right? Lower pH means more acid. And this causes a right shift in our oxygen dissociation curves, meaning that more oxygen is unloaded onto our tissues. Okay. This is also to say that hemoglobin and oxygen have a decreased affinity for each other. They become unattracted to each other, and it's easier to get that oxygen off of hemoglobin in the tissues. And this is considered a right shift. Things that can do this again are increased pH, right? So if you're, let's say you're in a metabolic acidotic state, this can be seen with diabetes, for example, when you're in ketoacidosis from extremely high sugars. This can also be seen if you are at high temperatures, if you have high CO2 levels in your blood, or we'll talk about a molecule called 2,3-DPG or 2,3-BPG, which can also mimic the Bohr effect or the right shift of our curve. But the main takeaway here that it's easier to get the oxygen off of CO2 because there's a lower attraction, lower affinity between oxygen and hemoglobin. The opposite of that is there's also your carbamino effect. Excuse me, this carbamino effect is another scenario causing a right shift. And this is where we have CO2 reacting with hemoglobin in order to produce carbamino hemoglobin. So excess levels of CO2 can actually bind to hemoglobin and produce this carbamino hemoglobin, which has a lower affinity as well between oxygen and hemoglobin. So this can be seen with increased metabolic activity, which just creates an environment where there's more CO2 circulating around. And then that can also increase the oxygen unloading in our active tissues, which really makes sense when you think about it. If you are in a state, let's say you're in a panic state, you have more CO2 building up in your blood, more of that CO2 can bind to hemoglobin, creating this intermediate carbamino hemoglobin, which makes it easier, again, to get the oxygen off of your hemoglobin, and you can easier unload it into those active tissues, which are undergoing an increased metabolic demand and would need that increased oxygen unloading because of increased CO2. So this is what we would see in increased metabolic states, which facilitates the easier offloading of oxygen in those tissues. Okay, now this is what I was describing earlier. So this is the shift or the curve that we described in its normal state. So we have our sigmoidal shaped curve. This is facilitating what we call cooperative binding, which means that this is the normal state of the oxygen and hemoglobin relationship. We have about 75% saturation in our systemic veins, and then 100% saturation in our systemic arteries. And this is the shape of the curve under normal circumstances. Now we can think about our curve shifting to the right or shifting to the left, which makes it either easier or harder to unload that oxygen in our tissues, depending upon the attraction, the affiliation between oxygen and hemoglobin. So if we were to shift our curve to the left, for example, this is where we have a higher affinity, higher attraction between oxygen and hemoglobin. Therefore, it's harder to unload or offload that oxygen, but it's easier to load it onto the hemoglobin. This can be seen with decreased body temperature or decreased altitude. Let's say you're somewhere that is below sea level, or this can be seen with decreased metabolic activity or metabolic demand. Right. The opposite of that is what we described in terms of the Bohr effect and the carbamino effect. Both of those are examples of a right shift. This is where there is an increased partial pressure of oxygen required to fully saturate your hemoglobin. So what we see here is a lower affinity, right? Oxygen and hemoglobin are not attracted to each other as much as they are with the left shift. So it's harder to load it, right, to get it on there, but easier to unload it because they're unattractive or have a lower affinity. This can be seen with the opposite types of scenarios. So increased body temperature, increased altitude. Let's say you're somewhere that's way above sea level. You're going to have higher partial pressures, and you're going to need higher partial pressures in order to get that oxygen onto your hemoglobin. This can also be seen with increased metabolic demand or exercise, increased exertional states, which is in alignment with that carbamino effect, right? With increased metabolic demand, you're making more CO2. That is creating the carbamino hemoglobin, which is what is an example of the right shift, where oxygen and hemoglobin are not very attracted to each other, right? It's harder to load, easier to unload, okay? And if you think about it, you don't really have to memorize these. Think about them in terms of a common sense approach. If you are at a higher altitude, if your oxygen and hemoglobin are not as attracted to each other, you're going to have to work harder in order to get that oxygen onto your hemoglobin, but you're going to have to work less to get it off and to unload it into the tissues, which makes sense. That is exactly what we want if you have a higher metabolic demand. We want it to be easier to offload the oxygen, but harder to get it on. Okay. Same thing with a decreased metabolic demand, right? If you have decreased metabolic activity in your tissues, it should be easier to get it on, but harder to get it off because it's not as demand in the tissue level. All right. So just kind of a simple sort of, logistic way to approach reasoning through this as opposed to just purely memorizing it. Okay. Another state that can mimic the right shift is the 2,3-DPG. This is also called 2,3-BPG. This stands for 2,3-bisphosphoglycerate or diphosphoglycerate, same thing. And this is a substance that is produced in your red blood cells under conditions of low oxygen, such as anemia, or if you go to a high altitude. So this is another state that can mimic a right shift, right? This is our third example of a state that can cause the right shift. the Bohr effect, right, which is if you have increased acid production or decreased pH, the carbamino effect, if you have more CO2 circulating in your blood, or the production of 2,3-BPG, which is also seen in states of anemia or high altitude. And what this creates is it inhibits oxyhemoglobin, right? The synthesis inhibited by oxyhemoglobin. And so this makes it easier to offload oxygen, which is what we want if we're at higher altitudes or we have anemia, right? We have a higher demand. Okay. So it enhances or increases the oxygen unloading, but it makes the loading a little bit more difficult in terms of the diffusion towards hemoglobin. The effect of carbon monoxide is another example of this. So carbon monoxide um, is a gas, very dangerous, very poisonous gas because hemoglobin actually has a greater attraction to carbon monoxide than it does for oxygen. And so what this does is this is a competitive binding. It prevents oxygen from binding onto hemoglobin, which can be fatal, right? Very, very dangerous. This is where we can have like carbon monoxide poisoning, which is fatal, right? This is where your hemoglobin is so saturated, it's completely saturated, it's kind of distracted, binding up all of this carbon monoxide, which it has a greater attraction for, and that it doesn't bind oxygen, which is what we need, for cellular respiration, et cetera, which is why it can be fatal. And this is just highlighting the right shift here that we described earlier. So 2,3-BPG produced by erythrocytes that are under duress, that need more oxygen, such as with anemia. And we can see how the curve itself shifts to the right here. We need higher partial pressures in order to fully saturate our hemoglobin, whereas in our normal scenario, we can saturate our hemoglobin with lower partial pressures. Okay, it's going to saturate easier, but it's going to offload harder with our normal curve. With the right shift, it's going to saturate, excuse me, it's going to be more difficult to saturate, but it's going to offload easier.